Yeah, hi, I'm Jeremy from the Riffle Lab and co-advised by Joe Thornton in the Department of Atmospheric Sciences. Yeah, thank you for attending my talk on how atmospheric oxidation impacts water navigation by hawkmoth pollinators. So animals use multiple sensory modes to navigate. Some examples include how moths use pheromone odor cues to locate mates, how cetaceans use echolocation to navigate underwater, and how migrating birds use vision and magnetoreception to navigate. So human activity is known to impact many of these navigation modes. So light pollution is known, for example, to impact animal navigation, as seen here in this classic scene of insects being drawn to lights at night, or hundreds of migrating birds being trapped in the New York City tribute lights before they kind of remedied that problem. And noise pollution is also known to impact navigation. In particular, sonar has been implicated in the unfortunate strandings of cetaceans. But the impact of pollution on olfactory navigation is less well understood. So common pollutants are detected by insects, and some pollutants have been shown to mask the detection of floral scents. And also the reactive components of air pollution, such as ozone and nitrogen oxides, have been shown to destroy floral scent compounds. So according to modeling studies, atmospheric oxidation has drastically reduced the transmission of some floral scent compounds in the atmosphere compared to pre-industrial times. For example, the transmission of osmine, which is one of the most common floral scent compounds, has been modeled to be reduced from a transmission of about 80% over a kilometer distance in pre-industrial times to 0% over a kilometer under present-day atmospheric pollution conditions. So my study system is the pale evening primrose Unathera pallida, which grows in the desert sands of eastern Washington. And it is a valuable resource for pollinators of that arid region. It's about in the center of the state of Washington. So Unathera pallida is visited by a rich diversity of pollinators, including diurnal butterflies, bees, syrphid flies, and nocturnal lasioglossum bees, hawk moths, other smaller moths, and some other unusual visitors like these Mavaleontids. And we are studying how atmospheric pollution impacts the reproductive success of Unathera pallida by affecting pollinator visitation rates. And so my experimental objectives are to determine the contribution of diurnal and nocturnal pollination to Unathera pallida fecundity, to determine which scent compounds the pollinators respond to and how they are degraded by pollutants, and to investigate how atmospheric oxidation of Unathera pallida scent affects pollinator visitations. I conducted pollinator exclusion treatments to determine the contribution of day and nighttime pollinators to fruit set in Unathera pallida flowers. And the results indicated that Unathera pallida requires pollination since fruit set was very low in the total exclusion treatment, which is about the same as in the self-pollination treatment. Well, yeah, both the day and night pollination alone resulted in significant fruit set. But excluding the nocturnal pollinators in this orange bar resulted in significantly lower fruit set than the no treatment control, which is here in blue. And this is kind of pollen supplementation. So Hyles lineata and some, which is this moth right here, and some Manduca species, which I'm pretty sure is Manduca quinquematulata, are the dominant nocturnal pollinators of Unathera pallida. And we're using Manduca sexta as a proxy for the few Manduca species in our lab experiments. I collected Unathera pallida floral scent samples using dynamic sorption by drawing scented air from bagged Unathera pallida flowers over these poropax scent traps. And then I eluted those scent traps with hexanes, and I analyzed the scent samples using GCMS to determine the chemical composition of the scent. And I also used GCEAD, or electroantenographic detection, using Hyles lineata and Manduca sexta antennae to determine which of the scent compounds are antennographically active. I then created a scent blend of these antennographically active compounds. And hawkmoths are active at night. So I'm studying the nighttime oxidative chemistry in the atmosphere, which involves nitrogen oxides and ozone, which are collectively known as NOx. And this is opposed to kind of the daytime reaction regime, which involves ozone and water vapor to create hydroxy radicals. So it's a different oxidative atmosphere in the daytime. So we use a chemical ionization time of flight mass spectrometer to analyze simulated atmospheric degradation of fluorescent in a glass flow tube. And so the results are that different fluorescent compounds were found to be differentially affected by ozone versus NOx oxidation. So monoterpenes like beta-pinene and osmine are most severely affected, while other compounds like this butanol oxime are not significantly affected. So the combination of ozone and nitrate radicals in NOx in here in the highlighted green sections degrades monoterpenes significantly more than ozone alone does. And next, we conducted no-choice behavioral assays in a wind tunnel towards fluorescent sources in order to determine the proportions of moths which would successfully locate the scent sources. And here we see Manduca sexta successfully navigating to the scent source and landing on the filter paper cone of the scent source. Next, we compared the moth responses towards my floral scent blend and scent blend that was oxidized with NOx. And the results showed that oxidation significantly reduced Hylie's lineata response towards my scent blend, while Manduca sexta response was basically unaffected. And so we then proceeded to compare the effects of the oxidation of undiluted versus diluted scent blend, and found that NOx oxidation of diluted scent blend yielded behaviorally significant results, whereas oxidation by ozone alone did not yield significant results. So this suggests that while Manduca sexta foraging behavior seems to be more resistant to the impacts of atmospheric oxidation than with Hylie's lineata, they might be susceptible to these impacts further downwind with scent dispersal and dilution. And so this is me setting up my field setup. So my field setup consists of glass flow tubes and a NOx generator for oxidizing the scent blend, which is then presented through filter paper cones. And mass flow controllers control air delivery from bottles of scent blend into these flow tubes. And there's a real floral scent treatment where I present the scent from one flower. And I use an air compressor connected to desiccants, activated carbon, and molecular sieves as my air source. And there is also a negative control here delivering just clean air. And then I set up infrared cameras and lights to record visitations to the paper cones of my different scent treatments. And also I set up this anemometer to measure temperature and wind velocity and turbulence, which can influence visitations. And then I'm setting up more cameras and lights. So the whole setup takes about an hour and a half after I streamlined my setup, although initially when I started doing it, it took like anywhere up to 5 hours to set up. And here we see a Hylie's lineata visiting the filter paper cone of one of my scent treatments in the field. And the results showed that my artificial scent blend results in a visitation rate which is not significantly different from real floral scent. And oxidation of my scent blend with NOx significantly decreases the visitation rate to a level that's not significantly different from my clean air control. Visits to real flowers are significantly higher than to all of my experimental treatments. All my treatments use humidified filter paper cones as artificial flowers compared to the real flowers, which have real petals and nectar and all. So in conclusion, we have shown that nocturnal pollinators are important for Uinifera pallida seed set, and that different floral scent compounds are differently affected by atmospheric oxidation. And this appears to differentially impact the different pollinators of Uinifera pallida in a lab setup and also in the field. And we suspect that this is due to like the alteration of the ratios of the different compounds in the floral scent, which can affect like recognition of the scent as a whole. And so next we plan to model the potential impacts of NOx pollution across the USA on hawkmoth pollination. It's kind of similar to what McFadrick et al. Did with floral scent transmission. And to do this, I created a chemical model of the degradation of floral scent chemicals in the presence of NOx by solving a set of differential equations of the rates of all of the chemical reactions that are occurring. And so I plan to integrate this with a map of NOx emissions across the US. And with that, I can model how hawkmoth recognition of floral scent is reduced across the USA. And yeah, I can do that by modeling kind of the distance over which we expect the moths to be able to recognize the scent blend, kind of similar to this plot here, which will be greater reduced the more severe the atmospheric pollution is. And so, yeah, I think that's all that I have. So I'd like to thank my funding sources, including grants from the NSF, NIH, FOSR, and WRF. I'd also like to thank the late Ben Hall, John Edwards, Robert Payne, as well as Lynn Ridderford and Jim Truman for establishing awards that funded my research. And yeah, I'd like to thank Jeff, wonderful Jeff, for all his advice, and the rest of my committee as well for their valuable input, and the Riffle and Thornton Labs for their support through my time here. Binyan for taking care of the insect colonies, and Sriram and my other undergrad researchers for their contributions to data collection and analysis. And yeah, with that, I'd like to